Okay, welcome to Let's Get to the Marks. Going through some questions from the 2024 Edexcel Paper 1 AS Biology. This one's on the circulatory system. And guess what? All of the content within this question I'm going over is relevant to you if you're doing OCR A-level biology or whether you're doing AQA A-level biology. It's all good. So expand your mind. Think outside of your syllabus, outside your box, and see if you can answer this using the knowledge you have on the circulatory system. Let's get into it. Don't forget to pause the video at the start of each question and then unpause and watch me walk through with the explanation. So, diagram one shows part of the internal structure of a human heart. Here we go. We got a bit labeled Q, a bit labeled P, S, and R. And we got some blood vessels coming on and joining to and from the heart. Point one. How many of the following statements about the human heart are correct? Structures P and S fill with deoxygenated blood when structures Q and R contract. Structure R receives blood from the pulmonary vein. When the cardiac muscle walls of P and S undergo diastole, blood leaves the heart and enters the aorta and the pulmonary artery. So let's get this all into the screen. And the answers are, the possible answers for this question are that none of these three statements are correct. One of these three statements is correct. Two of them are correct, or all three are correct. Well, let's go through and check it out. So I'm going to explain the answers now. Hopefully you've worked it out yourself. And let me zing up a little bit there to get it all into shot. So structures P and S fill with deoxygenated blood when Q and R contract. So when Q contracts, it's an atria, and it's going to send deoxygenated blood from the right atrium into the right ventricle here at P. Hopefully, you know that's the right atrium, right ventricle. And so that is true that when Q contracts, deoxygenated blood moves into P. However, in R, which is the left atria, when R contracts, that's not deoxygenated blood. That's blood that's returned to the heart from the lungs. So that is oxygenated blood. So we can say that this first statement is incorrect. Let's look at the next statement. Structure R receives blood from the pulmonary vein. So structure R is the left atrium, and the pulmonary vein, when you think about it, veins, and what you need to know off by heart is that veins always, without question, veins take blood towards the heart. And if it's coming back from the lungs, filled with oxygen, that's absolutely right. Oxygenated blood enters in here to R. So yes, structure R receives blood from the pulmonary vein. This is a true statement. Let's look at the third statement. When the cardiac muscle walls of P and S, so the ventricles here, this is our right ventricle, this is our left ventricle. So when these cardiac muscles undergo diastole. Now, what does diastole and systole mean? Diastole means relax. So when the right ventricle and left ventricle are relaxing, blood leaves the heart and enters the aorta and pulmonary artery. Well, if they relax, the blood isn't going to want to go up these blood vessels, is it? That's going to happen when they contract, when they squeeze. So actually, this is incorrect as well. So the correct answer here is B. Only one statement is correct, and that is the middle statement. All right, guys, we're going to move on to the next part of the question. Make sure that you pause, have a go at the question in your head or on a bit of paper, and then after that, you can unpause and watch the explanation. So the next part to the question is, complete the diagram to show the location of the semi-lunar valve in the pulmonary artery. Let's go up to the diagram and get this done. So we're looking for the semi-lunar valve in the pulmonary artery. So arteries go away from the heart. This is the right atrium. This is the right ventricle. This is deoxygenated blood. So deoxygenated blood needs to go to the lungs. So it's going to come up here, bus up there, and bus up there to the left and right lung. It's going to go red and come back into R through the pulmonary vein. So this is the pulmonary artery. Where are the semilunar valves going to be? Well, the semilunar valves, shaped like kind of moons, they're going to be sort of around there, anywhere inside this blood vessel, which is the pulmonary artery. So anywhere here is going to be where you draw your semilunar valves, and that is going to get you a mark. So well done if you work that out. Now we're going to go to the next part of the question. Describe the role of this semilunar valve in the cardiac cycle. So when it says describe the role of this semilunar valve, we're talking about the semilunar valve that is found within the pulmonary artery. Now, if we look at the semilunar valve in the pulmonary artery, See if you can come up with 3 bullet points. What is its job? When does it do this job? Pause the video, have a go, and then I'll go through it. So if we zing back up here, we're looking at if we have some valves here, don't worry about the direction of my valves or whatever. So if we got these valves here, what is their job? When the blood has gone past them, what do they prevent? They prevent the blood going back down here. They prevent blood going down here. Now, what do we call this area? They prevent blood from entering the good. So hopefully you know this. It's to prevent the backflow of blood from the pulmonary artery into the right ventricle. During what phase? Diastole or systole? It's going to be during um, during systole. They prevent the backflow of blood into the right ventricle. Let's get that all down, eh? Okay. Well, they're going to be open during systole, so we're going to say distole, I think, when it's relaxed. We don't want the blood falling back down in there. So, because those valves are going to be open. So, we can say they are open during ventricular systole, which means when the ventricles contract, they have to be open. Why do they have to be open? This allows blood to flow from right ventricle into the pulmonary artery and off on its journey to the lungs to get oxygenated. Now, when are they closed? They close during ventricular diastole. So during the relaxation of the ventricles, they're going to be closed. Why? This prevents backflow of blood into the right ventricle. So we don't want the blood to flow back. So if you put somewhere that probably the easiest mark is to say that valves prevent backflow, but then also to say that they'll be open during ventricular systole and they'll be closed during ventricular diastole. And that's going to get you all the marks. All right, let's get this off the screen and let's finish this question up. Hopefully you're doing all right on this. If not, you need to revise your circulatory system. So Part B, the diagrams show a cross-section of an aorta and a vena cava. So here's the aorta, here's the vena cava, really basic cross-section. We just cut one layer of cells, complete the diagram to show the relative sizes of the lumens. So what is the vena cava? You should be asking yourself that. Why don't you pause the video, have a go, see if you can work out which would have the bigger lumen. All right, the vena cava is a vein. It's the big vein that attaches to the right atrium and brings deoxygenated blood back to the right side of the heart. And of course, veins have low pressure. By the time the blood gets back to the heart, it's under low pressure. They have a large lumen. The aorta is probably where blood pressure is at its highest. It's right next to the left ventricle. It's connected to the left ventricle. So it's where the blood is being pumped to go around the entire body. And so it needs to be under high pressure. So relative to the thickness of the walls, it has a small lumen relative to the thickness of the walls. This is not a scale drawing. So you're looking at the lumen size relative, and they use that word relative, okay, to the size of the rest of the structure. So that will get us a mark. Of course, the aorta is an artery. I'm not going to say it's the biggest artery because I don't know whether it's bigger than the femoral artery, but we'll see. Which of the following would be in contact with blood in an undamaged vena cava? So if your vena cava is not damaged, what is going to be touching the blood? So look at this diagram. What is the red blood cells? I know it's a vein and it's deoxygenated, but what is going to be touching those red blood cells as it zings down there? It's the inside wall of the vein. And which of these is the name for the inside layer of cells in the vein and the inside layer of cells in the artery and in a capillary? What do we give the name of that? Is it collagen? Is it endothelial cells? Is it epithelial cells or smooth muscle cells? Well, it's actually endothelial cells. So the layer of cells on the inside of a blood vessel are known as the endothelial cells. Okay, that's the end of that question. Don't forget to hit the like, all that jazz. Let's get to the marks.
You're still here? It's over.
Okay, welcome to Let's Get to the Marks. Going through a nice, easy A-level biology genetic cross question. Don't worry about the exam board, it's suitable for all of you. It's an easy one, just one to warm up your day and get your revision started. Pause the video, have a go, then watch the walkthrough. The photograph shows the red, white, and pink snapdragon flowers. Plants with genotype FR have flowers with red petals, FW have white petals. We can see the lovely picture here. Deduce why heterozygous snapdragon plants produce flowers with pink petals. So, what do we know from looking at this? You can pause, have a go, 3 point answer. I'm going to explain it now. So, of course, you can see that we have FR. We're using capital letters, not lowercase. That it means it's dominant. Don't worry about my spelling, which isn't very dominant. It's very recessive spelling. Then we look at the FW, and this is for the white flowers. Again, it's a capital letter, so it's also dominant. We call this codominance or incomplete dominance. You can use either term. They're both correct. So we want to say the reason we get pink flowers is because of codominance. Boom, that gets you a mark. Now we need to explain that a little bit. Well, if you're heterozygous, you're going to have FR, FW, F capital R, F capital W, which means you're going to have one of each allele. You're going to have a red allele and a white allele, red and white allele if you're heterozygous. And they're both codominant, so both are expressed in the phenotype. So if you have FRFW, you're heterozygous, that's going to get you a mark. And both are expressed in the phenotype, will get you the third mark. But I can explain this a little bit further and say, well, both red and white are both going to be expressed. So we're going to get a kind of mix of red and white. We can call it an intermediate, something in between red and white, which will be pink. So pink is an intermediate or a mix of the red and white that are expressed in the phenotype. So 3 marks, codominance. If you're heterozygous, you have the FR and the FW gene, or allele, second mark, and both are expressed in the phenotype, third mark. And for a fourth bonus marker, if you miss one of the earlier points out, pink is an intermediate or a mix of the red and white that are both expressed. Right, let's move on. So, now we've got a little Punnett square, but it's a basic Punnett square, codominant monohybrid, 2 snapdragon plants with the genotype FRFW, so they're heterozygous, were crossed, determine the probability of each petal color in the offspring cross using a genetic diagram. So I know this is basic, I know this is easy, but we're using this one up as a nice warm-up for your day. So we're going to bust out in our Punnett square FRFW. Now that's going to get you a mark for writing the genotypes of the parents. We're now going to get the 4 genotype crosses for the offspring. So we're going to have FR for the first one, and then we're going to take the boxes and we're going to match them up with the relevant box. We're going to get FRFW there. Then we're going to go to the FW here and go to the FR. I'm going to write it as FRFW because I've done it there, and that's the heterozygous one. And then we're going to get FW in the 4th box. Now you're going to get your second mark for getting the correct genotype of the offspring. Now, looking at our Punnett square, we need to write down the probability in the table below. So how many reds are? Well, 1 in 4 is red. How many heteros are there? Well, there's 2 RWs. So we've got 2 pinks, right? 2 in 4. And how many whites are there? We got 1 in 4, right? WW. So now we're going to put that into a probability. I'm going to change it from a fraction to a decimal. We're going to go pink 0.5, red 0.25, white 0.25. Now let's look at the last part of this question. There were 416 plants given off as offspring from this cross. Predict how many of these flowers will have white petals. Well, you're going to do 0.25 times the total number of plants, 416. You're going to get 104. And that is our prediction of the offspring. Of course, you got random chance and fertilization of gametes, randomness of all of that business. So it might not end up like that. Anyway, guys, you know what I say. Sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks.
You're still here? It's over.
Welcome to Let's Get to the Marks, going through an A-level biology question. This one's on nucleic acids or nucleotides. Let's get into it. It's relevant to all exam boards, so don't be a little wuss and think, oh, it's not in my exam board. Yeah, well, how do you think they come up with those weird questions you've never seen before in your own exam board? They look at other exam boards. So you want to get the A star. Let's get into it. Nice and easy question, this one. Which labeled structure will contain the element phosphorus in this nucleotide? So which of these parts contains the element phosphorus? Elemental symbol P. Is it going to be the circle? Is it going to be the pentagon? Is it going to be the rectangle? Well, hopefully you know all of these off by heart by now. The circle is the phosphate group. The pentagon is the pentose sugar, which its specific name depends on which nucleotide we're dealing with. And then the rectangle is called a nitrogenous base. And you know they come in 5 flavors. You got your 4 for DNA, you got your adenine, thymine, guanine, cytosine, and then of course if you're RNA you have uracil instead of thymine. So that's just running through all of those. So which one of these contains the phosphorus? The correct answer here is the first letter, which is Q. And then we got some bonds. We got a phosphodiester bond and we just got a straight covalent bond here. So let's not worry about all of that. Let's zing down the answer there. Q, that'll get you 1 mark, and let's move on. Nice. You should know all of this stuff off by heart. If you don't know this yet and you're doing AES, make a revision card, draw that with the labels, and then on the back put the correct answers for each of the letters. Okay, part 2. Structure R is different in a DNA nucleotide compared with an RNA nucleotide. Name structure R in a DNA nucleotide and in an mRNA nucleotide. So let's go up and take a little zing and look at structure R. Right, it's got 5 points. It's a pentagon. It's a pentose sugar. That's the similarity. However, in DNA, it's called deoxyribose sugar, and it has 1 less oxygen atom than in when it's an mRNA nucleotide. The mRNA nucleotide does have oxygen in it, but it's got less, so it's known as a ribose sugar and not deoxyribose. So that will get you the mark. They're both pentose sugars. Anything with OSE at the end, lactose, glucose, fructose, they're sugars. Okay, which of the following bases would be found in a DNA nucleotide but not in RNA? I kind of went through that already. I mentioned it. Thymine is not found in RNA. You'd find thymine in DNA, but you'd find uracil in mRNA or tRNA. You wouldn't find thymine or rRNA. The diagram shows a polypeptide chain. We've got these little circles here which represent monomers, small molecules that join together to make a polymer. Number 2 says, first question, which row shows the correct type of monomer and the type of reaction that joins the monomers together? So what is the name of these monomers? Well, they're in a polypeptide. What's a polypeptide? Hopefully you remember it's a protein. And so they don't contain fatty acids. Let's get rid of those too. So the correct monomer is amino acid. How do we join 2 of these together? They're joined by a peptide bond in a reaction called a condensation reaction. All biological molecules are formed in a condensation reaction. All biological polymers and large molecules, they're joined together in condensation reactions. And hydrolysis, lysis means splitting or breaking. That's the breaking reaction. Which of the following types of bonds are involved in forming the tertiary structure of proteins? So we've got disulfide bridge, ester bond, ionic bond. Well, ester bonds are found in fats, so it's not that. We're talking about proteins here. Maybe that one, disulfide hydrogen ionic. Disulfide, we've got an ester bond again, that's in fats. And we've got a glycosidic bond, that's found in carbohydrates, not in proteins. So we can get rid of that. So our correct answer is B. And yeah, you've got disulfide bonds, hydrogen bonds, and ionic bonds, essentially covalent bonds, hydrogen bonds, ionic bonds. They form the 3D or tertiary structure of a protein. You can say hydrophilic and hydrophobic interactions as well, which also help form that tertiary structure of the protein, which is extremely important for its function. Right, last part to this question. Describe the role of RNA in the formation of this polypeptide. Essentially, guys, they're talking about how is mRNA and tRNA involved. Well, that's in translation, happens later, outside the nucleus. Transcription happens first. Translation happens later. They're asking you to describe translation here, guys. So see if you can put 4 bullet points on translation. Try to put 5 or 6 because we want to be specific as to the role of RNA in translation. Try to keep it focused on that. Pause the video, have a go, make it active. All right, unpause. I'm going to go through the answers. So I don't know why I say unpause, like you can hear me when you paused. Right, anyway, mRNA leaves nucleus via a nuclear pore. That's often the end of transcription, but we might start our bit about translation with that. Even if they say ignore, what does it matter? They're not taking marks off. So mRNA, what does it do then? It joins to the ribosome, right? Attaches to a ribosome. So that's always going to get you on a mark because we're talking about the focus of mRNA and tRNA. Then tRNA brings a specific, we need that word, amino acid dependent on the mRNA codon. And we can also say that the tRNA joins via its codon to the via its anticodon to the codon on mRNA. So the anticodon is on the tRNA and the codon is on mRNA, and they complementary base pair. That's definitely a mark, probably 2 marks. Something else which I haven't mentioned here, which would have got us a mark, is maybe potentially would have got us a mark. tRNA has a specific binding site that binds to the amino acid. So I didn't mention the binding site. You don't find that on mRNA. It's one of the differences between the two. tRNA has a binding site. Now, peptide bonds form between the amino acids as they're lined up by the tRNA, as they're brought down by the different tRNA molecules, the different amino acids line up and peptide bonds form. But is it the RNA that makes the peptide bonds? It's not. The RNA doesn't really have anything to do with the peptide bonds. So I would say that is an irrelevant point to the question. Would you lose a mark for that? No. So that's why I'm saying here, I would just write everything about translation and you'll pick up all the marks. At worst, they'll ignore this and you won't get a mark because it's not relevant to the role of RNA in translation in the formation of a polypeptide. So anyway, going back to the RNA, tRNA leaves, the first one leaves, I should say, and a second tRNA arrives, bringing a new amino acid. Now, it might be the same if the if the codon's the same on the mRNA, but and so which tRNA joins is dependent on the codons on the mRNA. So the sequence of codons or the order of the nucleotides on the mRNA, i.e. if it's CGG or GCG, whatever the order is of the nucleotides on the mRNA, that is going to determine our sequence or order of amino acids. And this forms the primary structure of the polypeptide. And of course, so it's the order of the amino acids that forms the order of the nucleotides that forms the sequence of amino acids in the primary structure of a protein. And then due to the different positions of the R groups, we're going to get different folding, different hydrogen bonding, giving us beta pleated or alpha coil. And then dependent on the R groups, we're going to get disulfide bridges and ionic interactions and things. And we're going to get that specific 3D structure. Anywho, that will get you all the marks. That's the end of the question. Here we go. We're on to question 6. This is question 6. Red blood cells contain hemoglobin. Oxygen molecules enter red blood cells and bind to hemoglobin. Glucose molecules can also enter red blood cells. The table gives information about these molecules. Glucose is a large polar molecule. Oxygen is a small non-polar molecule. Let's shrink that down so you can actually see what we're doing here. Describe how oxygen and glucose molecules can enter the red blood cells. Well, we have to think about what do they actually need to cross to get into the red blood cells. Why don't you pause the video, have a go on some paper, scrap paper, see if you can work out the 2 methods by which oxygen and glucose cross the membranes of the red blood cells to get in. Okay, I'm going to go through it with one of my crusty level 2 GCSE art diagrams. So these are my phospholipids. Of course, I'm drawing the cell membrane that would be found around the hemoglobin, the bilayer, and we'd need a protein channel as well. Now, oxygen is a small non-polar molecule like carbon dioxide. It can use simple diffusion to cross through the actual phospholipid bilayer. It doesn't need to go through a protein channel or a carrier protein. Glucose, on the other hand, has that delta negative oxygen, delta positive hydrogen. It's a charged molecule. It's polar. It has a dipole, and therefore glucose needs to go through a protein channel or intrinsic protein, a protein that runs all the way through the cell membrane. And so glucose enters by a form of facilitated diffusion. It's actually co-transport involving sodium ions, but we don't need to go into that kind of depth here. So we can just put oxygen can enter, I can't even speak properly. Oxygen can enter the cell by simple diffusion from a high concentration outside the cell to a low concentration inside the cell through the phospholipid bilayer. Whereas glucose needs to enter the cell as it's a polar molecule. It cannot cross the bilayer as it's not soluble in the bilayer. Therefore, glucose must pass through a protein channel by facilitated diffusion. By the way, this is also from high to low concentration, or you can just say down a concentration gradient, which is always a little bit easier if you memorize that term rather than having to say travels from high to low. And here we go. That's going to get you both the marks, which is kind of nice. Remember, protein channels are used for facilitated diffusion, and generally we talk about protein carriers as what is used for active transport. And although glucose entering cells sometimes in certain parts of the body involves active transport, it's indirect. So anyway, enough of that whiffle waffle. Let's get on with B. Collagen and hemoglobin are proteins associated with the cardiovascular system. Give 3 differences between the structures of collagen and hemoglobin. So there's quite a lot of differences here we can go through. Okay, why don't you pause the video, have a go on some scrap paper, see how many marks you get out, 3, or you just do it in your head. Okay, so it says give 3 differences between the structures of collagen and hemoglobin. Collagen is really a protein that's used for strength and structural its structural properties. Hemoglobin is a globular protein. So hemoglobin is actually um, globular. And what do we mean by that? It's a sphere. So we want to say that hemoglobin is not a circle, that's 2D, but a sphere. And leading on from that, we can say whereas collagen is a chain. Um, so it's in straight chains with cross links between the chains. It's for strength. And on that, we can also say collagen's made of 1 polypeptide, whereas hemoglobin's made of 4 polypeptides, so multiple polypeptides, so it has a quaternary structure. I'll come back to that. Hemoglobin also, because it's globular, has hydrophilic groups on the outside, water-loving groups that can form hydrogen bonds with water. Hence, hemoglobin is soluble, whereas collagen has hydrophobic groups on its outside, which repel water, so it's insoluble. Now, what was I saying about the quaternary structure? Yes, collagen's made of 1 polypeptide chain, hemoglobin's made of 4, so it has a quaternary structure. Now, having a quaternary structure, that gives you your final 3D shape. And yeah, so that's another difference between the two structures. Okay, and another point you might remember that hemoglobin has Fe2 plus iron ions inside it. These are called prosthetic groups. They're actually a type of cofactor that are tightly bound within the polypeptide chains. They're what attract the oxygen in hemoglobin to form oxyhemoglobin. And you don't find these prosthetic groups in collagen. Okay, hemolysis is the breaking open of a red blood cell, causing the release of hemoglobin. The effect of concentration of 3 different alcohols on the hemolysis of red blood cells was investigated. Samples of the red blood cells were treated with different concentrations of 3 alcohols. The table below shows the formula of the alcohols. We've got ethanol, propanol, butanol. A microscope was used to count the number of intact red blood cells. The investigation was repeated twice. The mean percentage of red blood cells that had hemolyzed was calculated and the results were shown in the graph. Okay, so we can see we've got percentage of red blood cells hemolyzed, which means split open, and we've got them for butanol, propanol, and ethanol, and we've got the changing alcohol concentration along our x-axis to show the patterns. All right, let's go down and see what the actual question is. State and justify a suitable control for the investigation. It's worth 2 marks. So we're looking at what is a control? Not a control variable, but what should we use a control? Well, we're using alcohol, right? And we've got different concentrations of alcohol, butanol, ethanol, propanol, and we're looking at hemolysis with the different concentrations of alcohol. Well, of course, our control should be replace the alcohol with distilled water and see is it the actual alcohol that's causing the hemolysis, or does hemolysis happen with water when there's no alcohol? So yeah, your control variable is normally what you're using to compare your independent variable and see if it has actually had an effect on the dependent variable. So in this case, we're going to use water to see is it the actual alcohol that's having the effect on hemolysis, or would the same effect happen with water? Okay, evaluate the effect of concentration of the 3 alcohols on the hemolysis of the red blood cells. So we want to look at the 3 different alcohols. Do they have an effect? Does it matter what alcohol you're using or to the percentage hemolysis? And does it matter what percentage, what concentration of the alcohol you use to the amount of hemolysis? So well, we can look at this as we go from ethanol to propanol to butanol, the chain length of the alcohol gets bigger. So we go from 2 to 3 to 4 carbons in the chain. And we can see that as we swap from ethanol to propanol to butanol, we get more hemolysis, more red blood cells bursting open. So the chain length or the size of the alcohol is a definite factor. We can also see that it also we could say which of them has the most hemolysis. So that would be butanol had the most hemolysis. Can't really read that there, can you? And ethanol has the least hemolysis. Let me change to pink, make it a little bit more visible. So we can also see that. And we can also see that with all three alcohols, doesn't matter whether it was ethanol, propanol, butanol, as you increase their concentration, you increase the amount of hemolysis as well. Okay, now this was an evaluation. So we've said three or four like positive things about the trend shown by this evaluation. And we've used the graph to back that up. We now want to say some things that maybe make the investigation question the validity of the investigation. So is it reliable? It looks like it was only repeated twice. That's what they said in the stem of the question. They repeated it twice before taking a mean. Maybe they should have repeated it 3, 4, 5 times so they can spot anomalous results more easily. And did they control all the other variables that might affect the permeability or the hemolytic nature or when red blood cells burst? Did they control everything else that could affect the membrane? So what other things affect the permeability or the flexibility of the membrane? Of course, temperature. They should have used a water bath, and that would have stopped it melting, the phospholipids melting, and things like that. Kept the volume the same of the alcohol. They didn't really talk about keeping the volume the same. Scientists have investigated the functions of DNA polymerase, described the function of DNA polymerase in producing a polynucleotide. So we're talking about semi-conservative replication. You might make the mistake and think we're talking about polypeptide. Why don't you pause the video and have a go? Okay, so DNA polymerase is not used in protein synthesis. It's used in semi-conservative replication of DNA in the formation of another polynucleotide. And what happens is the parent strands split apart, as I've shown here. And then we get these DNA-free nucleotides line up by complementary base pairing to the template strand, which is my pink one here, and my green little dashes are my free DNA nucleotides. And essentially they pair up. Now, what is the job of the DNA polymerase? Of course, the DNA polymerase attaches at the start codon or the start of the gene and works its way down the strand, forming the phosphodiester bonds. And what type of reaction does it do? In order to form the phosphodiester bonds, which join the DNA-free nucleotides together, what type of reaction does it do? What's the making reaction in biology? It's condensation. So really, the key points here is that DNA polymerase joins the DNA-free nucleotides together by forming phosphodiester bonds by a condensation reaction. And that's going to get you all the marks. So really, DNA polymerase moves down, forming the phosphodiester bonds by condensation reactions to make a sugar phosphate backbone, or to join together the DNA-free nucleotides. You might want to say to join together those. Okay, let's look at the second part of this question. Scientists have discovered a property of DNA polymerase that detects and corrects errors in DNA replication. Discuss the possible benefits of this property of DNA polymerase. 4 marks. Now, this might seem really obvious, but yeah, let's get into it. Pause the video and have a go, guys. All right. So if the DNA polymerase can detect errors and correct them, it means that you're going to get less errors in the base sequence, right? You're going to get less errors in the base sequence that's constructed. And this means if you have less errors in the new strand, the green strand that we've got above there of DNA, then that means less errors will be transferred to the mRNA during protein synthesis with RNA polymerase. And it means that you're more likely to get the correct functional protein with the correct primary structure and hence the correct tertiary structure for an enzyme, for example, the correct active site to form um, an enzyme substrate complex, that kind of thing. And if you get non-functional proteins that can't do their job, this could lead to cancer. So it means we get less chance of cancer. Okay. And that's due to sort of proteins that have a role in inhibition and stimulation of mitosis. Some people have DNA polymerase that cannot detect and correct errors in DNA replication. These people have an increased risk of developing cancer. The increased risk of developing cancer occurs when a person inherits two faulty recessive alleles of DNA polymerase gene. The pedigree diagram shows the development of cancer in one family. We've got a key over here. Individuals A and B have a child. Explain why they may choose pre-implantation genetic diagnosis, PGD. Pause. Have a go. All right. Well, we know for a fact that one of the parents contains both the recessive alleles because we see parent B is a female with cancer. She has both of the alleles, the recessive alleles, um, for um, the faulty DNA polymerase gene. So she has those alleles. This means that the child's going to inherit one of them, and the dad also, because they've had a child already with cancer, this means that the dad also possibly has that allele. So there is a probability that the child will get that. Some of the children already have cancer, so they know that it could be that the mum has these recessive alleles and the dad has one of them, maybe he's a carrier, and so there's a probability that their children will get it. So having PGD would allow them to be better informed about what to do with the embryo if it contains those alleles. Walk through. Right, question 6. The effect of enzyme concentration on the rate of reaction was investigated. Starch is a polysaccharide. Which of the following statements about polysaccharides are correct? Pause the video, have a go. All right, starch contains amylose, a straight chain containing 1 to 6 glycosidic bonds. Glycogen contains amylopectin, a branch chain containing 1 to 4 and 1 to 6 glycosidic bonds. Polysaccharides contain 1 to 4 and 1 to 6 glycosidic bonds, allow a rapid release of energy, and starch and glycogen are both soluble in water. Okay, so we can go through and cross out the ones that we think are definitely wrong. So does starch contain amylose? Yes, starch does contain amylose. Can you remember the two different types of starch? You have amylose. Don't ask me why I'm writing in pink. And amylopectin. And what's the difference? Well, amylose is a straight chain. So amylose is a straight chain. So that's correct. And starch does contain amylose. Amylopectin is the branch chain, but they're asking us about amylose here. Now, if it's straight chain, what type of bonds would it have? 1 to 6? No, it'd have 1 to 4 glycosidic bonds. 1 to 6 glycosidic bonds occur at the branches. So that's an incorrect part to that. So A is not the right answer. Glycogen contains amylopectin. Amylopectin's found in starch. It's not part of glycogen. They're both heavily branched and they contain 1 to 4 and 1 to 6. So those two facts are right. They are branched and they contain 1 to 4, and if they're branched, they have to have 1 to 6. But amylopectin is not in glycogen. Polysaccharides containing 1 to 4 and 1 to 6 allow a rapid release of energy. Well, yes, that is true. Everything here is correct. I don't know about if I'd say rapid, but yeah, a more rapid release of glucose in order for respiration. That's true. Starch and glycogen are both soluble. No, they're both insoluble. Otherwise, our liver would explode by all the water going in by osmosis, as it would lower the water potential. And same with starch. We don't want our big storage molecules to lower the water potential and have an osmotic effect. Okay, part B, amylase breaks down starch into maltose. The diagram shows the effect of adding iodine to flasks containing different concentrations of starch. You can see it starts off yellow-brown, and then when there's a lot of starch, it will go blue-black, but it's kind of changes in between. The effect of amylase concentration on the rate of reaction at 2 different temperatures was investigated. The table shows the results of this investigation. We've got rate of reaction. AU stands for arbitrary units, sorry. Rate of reaction at 45 degrees. All right, they've done 2 different temperatures and they've changed the concentration as well. Okay. Explain how to carry out a valid investigation to collect the data shown in that table above. This is quite a common question. They'll often give you a graph and say, how did they carry out a valid investigation to find this data? So I would practice these questions. These are quite common, and you need to be able to study a table or study a graph and then say, oh, how did they get the data? So you want to explain how they changed the independent variable. We need to explain how they collected the dependent variable, what they measured. We need to then think about what they controlled and then say, yeah, and that's it really. Just explain how they got all this data. So look, what was their independent variable? It's found in the first column. You might want to pause the video and have a go at this, but these ones do take a bit of practice, and there is an exam technique to these, and you can learn the pattern and then just repeat it every time you get this question. So first of all, let's explain how they got the independent variable. They must have used different concentrations of enzyme, right, between 1 to 8 millimole per decimeter. How did they get the zero enzyme? Also, they must have used distilled water to get the 0 millimoles per decimeter. So we've just explained that they used between 1 and 8 millimole per decimeter concentration of the enzyme amylase. They made a solution. Was it amylase or maltose? Whatever, maltase. I can't even remember what the blooming thing was now. Very good. So I think it was amylase. Anyway, we can just call it the enzyme, as it says so in the table. So yeah, 1 to 8 and distilled water to get the 0. Let's zing back down. All right, they added iodine. So if you look up here, you can see no starch. It was yellow brown. So what they then did is they added iodine. The enzyme was amylase, obviously. Oh, I forgot to put 8 here. Excuse me while I fix this up, guys. So let's put 8 in there. Okay, what else? What else? Right. Now you could make up a volume. They added 20 centimeters each time, or they added the same volume each time. So we're going to say what they controlled to get valid results, because they use the word valid. So they want us to say how to keep the control variables the same, how they must have done it. So they might have kept the temperature the same, because that would affect the rate of reaction, using a water bath. And what do I mean by that? Because they've changed the temperature to 30 and 45. But I mean, when they were doing it at 30, they must have used the water bath to keep it at 30, so it didn't go to 31 or 32. Right, they must have added a pH buffer, right? Because change in pH could denature the amylase, so maybe they added a pH buffer to the solution to prevent pH changing during the reaction. All right. Now, how did they actually calculate the rate of reaction? We have to think about how they did that. So, how did they work out that the rate of reaction in AU was 31 or 41? So, I think what they did is they keep the starch concentration the same each time as well. That's another control variable. So keep the starch concentration the same each time, because they're changing the enzyme, the amylase, and keep the volume of the starch the same each time. And then they're going to add, we probably should say 2 drops of iodine each time, or state a specific amount, not just, I've put just add iodine. You should really say add iodine in potassium iodide solution, add like 2 drops. And this is going to make it go blue-black if there is starch present, right? If the enzyme has broken it down, we're going to time how long it takes for it to go yellow-brown, or to stay yellow-brown, not to change blue-black. And in fact, the best way to do this is to get like a well, to get I like a spotting well with little holes in it, little dips, little wells in it, and you put a drop of iodine in each one of the wells, and then you take a sample from your solution that's reacting, your amylase and starch that's reacting. You take a sample like every a set interval, like every minute. No, every minute would be too long. I reckon take a sample every 10 seconds, maybe. Every minute? No, a minute would be too long, because how would you know if it's all broken down between 2 and 3 minutes? You wouldn't know where. So probably best to make those time intervals a little bit smaller. So I'll change that in a second to 10 seconds. But you then want to take a sample like every 10 seconds, add it to the well of your enzyme and substrate solution. And if it goes blue-black, it means there's starch still there. If it stays yellow-brown and it doesn't change, it means all the starch is broken down. So we should be able to narrow it down to within 10 seconds, if we're doing time intervals of 10 seconds, to the time it took to break it down. And then as they've calculated this in arbitrary units, we could do the rate as 1 over time. Essentially, we could do the rate as 1 over time. And that would give us the rate. And I think that's going to get us all of the marks, right? Oh, actually, I forgot. They should, in order to get valid results for each of the different enzyme concentrations, they should repeat like 3 times and then calculate the mean time for it to stay yellow brown. And that way you can check for anomalies, and your results are going to be more reliable. So, yeah, should add that in as well. Okay, the last part to this question says, comment on the effects of enzyme concentration and temperature in this investigation. So what we want to do is we want to go back up and look at the results, and we want to look what's happened to the rate of reaction as the enzyme concentration increases. Well, we can see at 30 degrees, the rate of reaction increases. At 45 degrees, the rate of reaction also increases, but we can also then compare the temperatures and say, but however, at every enzyme concentration at 30 degrees is greater than the rate of reaction at 45 degrees. And then we need to put some sort of explanation for that. So my thing is, as you increase the enzyme concentration, there are more enzyme particles. You get a greater frequency of collisions between enzymes and the substrate. More enzyme substrate complexes form. That's as you increase the concentration, and that's for both 30 and 45 degrees. You're going to get that increase. However, at the concentrations of 7 and 8 millimole, we saw with the 30 degrees that it stayed, the rate of reaction went to 100 and didn't go more than 100. So it's got to its maximum at that point. It didn't matter. Increasing the amount of enzyme couldn't further increase the rate of reaction, because at that point, the substrate is a limiting factor and there's not enough substrate. So what we want to do there is also compare the column of 30 degrees to 45. Now at 45 degrees, we might think, hey, more kinetic energy, should be more enzyme substrate complexes forming because they're moving faster, more frequent collisions. However, what's probably happened is the enzymes have started to denature. And at that point, we want to say the enzymes have denatured, the active site has changed shape. The active site will no longer form an enzyme substrate complex, or is no longer complementary to the substrate. So that's why we have that lower rate of reaction at the higher temperature. And that will be our explanation for that. So when you're asked to comment, and the key word here is comment, you need to describe and explain. So it's kind of like saying describe and explain. So it'd be nice. Okay, let's get on with question 7. Obesity indicators such as body mass index, BMI, can be used to assess the risk of developing coronary heart disease. The table shows categories of BMI. A man has a body mass of 65 kilograms. He has a normal BMI. Calculate the minimum and maximum possible heights for this man in meters. Use the formula BMI equals mass divided by height in meters squared. Calculate their minimum height and their maximum height. So why don't you pause the video now, grab your calculator, pen and paper, and have a go at this one. Okay, so what we need to do is we need to look at the normal range. You can see it's 20 to 24.9. So let's work out what the maximum and minimum possible heights are for this man in meters, where the BMI is 20 at its lowest, and at its highest, it's 24.9. So to rearrange this, I'm going to times both sides by the height squared. That's going to put my height squared on the left with the BMI equals mass. But I want to get the height in meters squared on its own. So I'm then going to divide both sides by the BMI. So I'll have height squared equals mass over BMI. So first of all, I transfer the height squared over to where the BMI is on the left, but then I need to bring the BMI over to the right. Now I need to square root both sides, not square both sides. Sorry, I should have said square root both sides. And that will leave me with height, because by square rooting height squared, you'll just get height. And that will leave me with height equals mass divided by the BMI, the square root of the mass divided by the BMI. So I want to do 65 kilos over 20, and then I want to square root that, and that is actually going to give me the highest height. It's the lowest BMI, but for that mass, it should give me the tallest person, because obviously their BMI is lower because they're so tall, and it's 1.8 meters. Now what we need to do is work this out for 24.9. So the exact same system. So we're going to do 65 kilos because their mass does not change. But now we're going to do with a BMI of 24.9, and we're going to square root it for the same working out we did before, because we're trying to find height. And this is going to give us the height. So 65 kilograms divided by 24.9, and then square root your answer in your calculator. And when you put all of that in, you are going to get about 1.61, I don't know, 5, 6. So we're going to round that to 1.62 meters. And that is going to be the lowest possible height, or the minimum. Maybe we should call that the minimum. Seems like a lot of work for 2 marks, but that's going to get you both of them. So let's move on. Okay, one risk factor for coronary heart disease, CHD, is obesity. The table shows the incidence of CHD for people with a differing number of risk factors. Describe the relationship between the number of additional risk factors and the incidence of CHD. This is an easy one. I've kind of jumped the gun. You should have paused the video and had a go. But the general relationship is that as additional risk factors increase, when you're given a description, just give the basic general description. The incidence of CHD per 100,000 people increases. Now, pause the video for this next one. The second mark is always hard in these descriptions. You've got to give some nuance. So what we want to say here is, aha, yes, it's increasing, but look, between 1 and 2 additional risk factors, it goes up by, I don't know, it goes up by, well, more than 50%, but now it goes up by like 200 and something percent when we go from 2 to 3. So we want to say the increase from 2 to 3 additional risk factors has a massive, or a very big increase in the incidence of CHD per 100 people. We could do some sort of calculation to show that 615 divided by 210. There's a 2.93 times increase when we go from 2 to 3 additional risk factors. Okay, let's go. State one risk factor for CHD other than obesity. Now, there's a whole load of these. Be aware, guys, you can easily think of one of these. You should pause now and have a go. Okay, be aware, guys, that sometimes you want to talk about a lifestyle factor, and that is like an environmental factor, so you can't include age, gender, or genetics in that. But here they just say state one risk factor for CHD other than obesity. So they haven't specified that it's a lifestyle factor. So we could really say anything that increases your blood pressure. Now, high blood pressure can be genetic. High blood pressure can be due to your age. It increases with age, and gender can affect your blood pressure as well. And then you've got other factors that affect the blood pressure. So anything with a high blood pressure is going to damage that endothelium of the in the lumen of the artery and increase the chance of buildup of plaque and CHD. So those were all the different risk factors, and you only had to pick one. The table shows the number of deaths due to CHD in 3 countries. You've got England, France, Russia. Number of deaths per 100,000. We've got men and women. Plot these data in a suitable graphical form. It's worth 4 marks, and this looks quite easy because we can see we've got categoric data here with England, France, and Russia. So what we can do here is a bar chart, because our independent variable is categoric, and therefore we're going to bust out a bar chart. And what you're going to get the marks for here, they've left the axes for you to do. So you need to use over half the graph paper to score the mark for the scale on the axes. And then you need to label with the correct units. Obviously, the units for the independent variable are just countries. So we're going to do England, France, and Russia, and I'm using like 4 centimeters for each, roughly. This is gonna involve me piddling around to try and get this all in space, guys, on here. Okay. Um, Jeez Louise. Now, on our y-axis, which is the one running up the side, sorry, I'm just shortening all these boxes, but on our y-axis running up the side, we're going to do number of deaths per 100,000. Again, I'm going to have to mess around to try and get this. How would you get this? How would you write this on in your actual exam when you're not allowed to write over that margin? How would you fit that in? I've got no idea. I guess what we would do is we would write that on the graph paper and maybe make our axes. Yeah. What I should have done is made my y-axis maybe 2 centimeters in, but I haven't done that now, so I'm not going to bother. So look, in the exam, I'd be in trouble here, and I may lose a mark because I've written number of deaths per 100,000 over the graph, where it says do not write in this area. I'm going to have to shift it onto the do not write in this area. I'm just going to have to take that one, guys. Okay. All right, so what I want to do is just get these names down here off of the graph paper, And my biggest number is 700 in the number of incidents of CHD per 100,000. So my y-axis must go up to 700, which is quite a nice number because I can use 2 centimeters or two of the kind of big squares for each 100. You'll see 700. I finished just before the top, so absolutely nice. I just got to rearrange. I just got to arrange this. It's going to take me a little bit of time, a little pain in the butt because I've got to move these numbers over so they're not in the way when I plot the data. Um, yeah, that's a pain. Okay, bear with me as I squibble, squabble, squabble, and get this all in the right place. And then we're going to start plotting the data on. So what we're going to do, this is going to be a bar chart. How do I know it's a bar chart? Well, England, France, Russia. So it's categoric. So it has to be a bar chart. Now, you might say, well, how are you going to do male and female? Well, I'm going to put the bars directly next to each other so we can compare them. I'm writing F. Oh gosh, I should have written male and female. I've written a W for women. That is a woman. That's a bit caveman-like, but never mind. Okay, sorry about that. Yeah, anyway, I'm going to stick with that. So we've got male and then female. Of course, I've stupidly put a W. You'd want to put a key explaining what the different color bars are or what those letters stand for. The examiner isn't just going to take you writing M and F. So if you explain that with a key. Now, ideally on my x-axis under England, I could have written male and female or M and F just above England and just above France. So I don't have to write it actually on the bar, but I'm being a bit crude. And there you go. That's your bar. And that's going to get you 4 beautiful marks. As I say, I recommend writing male and female on the x-axis or making a key and shading them in slightly differently and then making a key to explain what is male or female, and that will be acceptable. Okay, let's move on. Atherosclerosis is a type of cardiovascular disease, CVD, that causes narrowing of coronary arteries. How many of the following types of medication would reduce the risk of CVD? We've got anticoagulants, hypertensives, and statins. Pause the video. Have a go, folks. All right, so if we think about, I'm going through the explanation if you hadn't guessed. Anticoagulants, they make the blood less viscous, they make it thinner, and then you're less likely to get a blood clot, right, if your blood is not so thick. Antihypertensives, now, very complex system, but these essentially lower your blood pressure. And so if you have a lower blood pressure, there's less damage to the inner lining of your artery, the inner wall. And then statins will lower cholesterol, so less chance of fatty plaque, fatty deposits and plaque building up. So I'd say all three here. If you went for all three, you'd be absolutely correct. Okay, let's check the next bit. The diagram shows a procedure called balloon angioplasty. Balloon angioplasty can reduce the immediate risk of a person having a heart attack. It will not prevent further narrowing of the coronary arteries due to atherosclerosis. We've got a guiding catheter, narrowed artery. We've got a balloon catheter with an uninflated balloon. Then they inflate the balloon and you have an unblocked artery. It's not permanent, but it will in the short term give you some relief. Explain why the balloon angioplasty could reduce the immediate risk of a person having a heart attack. Pause the video, have a go. All right, these come up all the time, and it's a really common 4 bullet point. So you should have this off by heart on a revision card. So anytime we're trying to make the heart work better or reduce CHD or CVD, we want to increase blood flow to the heart muscle. So we make a bigger space in the coronary artery, we increase the blood flow to the heart muscle. This means an increased supply of oxygen and glucose reach the heart muscle. If you have more oxygen and glucose, what can those muscle cells in the heart do more of? They can allow more aerobic respiration, and then we're going to get greater muscle contraction, and the heart cells aren't going to die, essentially. So if you don't, and again, you could be asked about how a reduced blood flow from fatty deposits damages the heart, and again, you would follow the same kind of course. Lovely. Essentially more ATP produced is going to mean greater muscle contraction, etc. So this is all good. It's going to reduce the risk of heart attack. Right. Some organisms, we're on question 8, do not have a circulatory system. The photograph shows a nematode, not even going to say that, as seen with the electron microscope. We've got a magnification of times 1300. Calculate the actual width of the nematode from X to Y, 2 marks. Boom. Why don't you have a go? Okay, hopefully you remember this. It's the I am equation. You've got image over actual times mag. Now, we need to check out what X and Y is. Check out the level 2 GCSE art ruler drawing there. So if you were to measure this with your ruler, you're going to get something like 13 millimeters. There is a range that they're going to find acceptable. Okay, so it's going to be roughly 13 millimeters between X and Y, and that is the image size. When you're doing with microscopic data, that's not going to be the actual, and we're finding the actual, so we're going to do I over A. Oh, sorry, I over M equals A. So we're going to go 13 divided by 1300. and that should give us 0.01, but this is in millimeters. Now check the answer. It's supposed to be in micrometers, so we need to times by 1,000 to get it into micrometers, and it's 10 micrometers. Obviously there'll be a range between like 9 and 11 that they'll allow based on your measurement with the ruler. Explain why this nematode does not need a circulatory system. Pause, have a go. Okay, it's a very small organism, therefore it has a large surface area to volume ratio, which will allow the rapid diffusion of substances. So it's able to meet metabolic demands, i.e., get enough glucose and oxygen for respiration by diffusion alone, enough amino acids for making proteins, etc. You could also talk about thin walls, short diffusion pathway. There are other ways you could go down, but I went for the small organism, large surface area to volume ratio. Right, B, humans have a circulatory system. The graph shows the change in blood pressure in different human blood vessels. So we got blood pressure in millimeters, Hg. Hg is like mercury. We've got aorta, arteries. We've got arterioles, capillaries, venules, medium and large veins, and vena cava going along the x-axis here. Explain why the blood pressure increases and decreases in the aorta. So why does the blood pressure increase and decrease in the aorta? So essentially, during ventricular systole, keyword, we have contraction of the left ventricle, which pumps blood into the aorta, increasing the blood pressure in the aorta. They go through the semilunar valves, which open when there's a high pressure underneath, and we end up with a high pressure in the aorta. Why does it decrease? Well, that's when we're going to go to diastole, and the ventricle is going to be relaxed. The semilunar valves are closed. So there's no new blood at this time entering the aorta, and obviously the blood that was in the aorta has left on its journey around the body. So we get a drop in blood pressure. And there we go. Okay, calculate the percentage decrease in blood pressure as blood travels through the capillaries. Give your answer to two significant figures. We need to go up to the graph to have a look at this. Humans have a circulatory system. Let's zoom in. Okay, so what are we looking between? We are going in between when the blood first enters the capillaries. What is the blood pressure when they enter the capillaries and leave the capillaries? So, oh God, I've got to work out what each square is. Each square is, let's see, 20 divided by 5. Okay, each square is worth 4. So that's going to be 36 millimeter Hg. And then capillaries, this is a really bent line. But it's looking like 16, but actually let's move it down. It's 15. It's really in between a square. So it's about 15 mercury there, or 13. Sorry, because each square is 4. So 20 minus that, minus 2. Yeah, I don't know about 13. I think that's more likely. 16 plus 2. Oh, it's more likely 14. I think I'm out there. I think that's supposed to be 14. Anywho, what you're going to do to work out the percentage difference, you're going to do the change over the original times 100. So I'm doing 36 minus 13 times 100. Now, I haven't checked the mark scheme on this one. That 13 millimeter Hg actually might be 14. The accuracy of my line is not great, but here we go. So I'm going to do 23 because that's the change in the blood pressure divided by the original blood pressure of 36 times 100, and I got 63.88%. Probably going to do this to 63.9, and then that's if you want to do it to three significant figures, you're going to only get 1 mark because these guys wanted it to 2 sig fig. So we're going to 64%. So the correct answer is 64%. I think that's right. I think it must have been 13. Maybe my eyes are just a bit biggly boggly. Right. The squid is an invertebrate animal that lives in the sea. The photograph shows a squid. The diagram shows the circulatory system of a squid. The gills contain gas exchange surfaces. Okay. Compare and contrast the circulatory systems of a squid and a lumen. And a human and a lumen. Oh my gosh. Compare and contrast the circulatory systems of a squid and a human. So compare and contrast, you need to translate those words if you're an Edexcel biologist. Compare means what's the same, contrast means what's different, and you're basically just describing what's the same and what's different. Three things. So two similarities, one difference, going to get you to 3 marks, or vice versa. And there's going to be about 6 or 7 things here. So humans have a systemic heart, so do squids. Um, the human heart has 4 chambers. The squid systemic heart only has 3 chambers. That's a difference. There's loads of differences here, aren't there? It looks like a squid has 3 hearts. A human only has 1 heart because there's 2 branchial hearts as well. The squid has, which are like kind of their version of our right atrium that pumps to the lungs. That pumps there to their gills to get oxygenated. Of course, we don't have gills. We have lungs. Um, yeah, the squid has 3 separate hearts. What else can we say? What else can we say? We can talk about we've already mentioned the chambers, so that's 2 differences. What about similarities? They're both closed, right? So the circulatory system is a closed system, which means the blood is encapsulated within arteries, capillaries, and veins in blood vessels. It's not like in an insect where it is, the it circulates without blood in blood vessels. It's like free flowing between the cells in insects. So they have an open circulatory system. So yeah, closed circulatory system would be a similarity. Both contain, and you could name as well any blood vessel, like i.e. squids and humans both have veins. Squids and humans both have arteries. Squids and humans both have a capillary network. There's kind of tons of points you could go over here. You should be able to get these quite easily. There's quite a few points. Let's move on to the final bit of this paper. At last, the student wished to investigate the effect of caffeine on the heart rate of some vertebrates and invertebrate organisms. Describe 2 potential ethical issues which should be considered before selection of the organisms to include in this investigation. So, pause the video, have a go. Nice and easy ethical one. So, what do we need to think about? We need to think about the pain that the organism is going to undergo. Invertebrates don't have a nervous system, so we need to think that maybe they don't feel pain at all, or maybe they will feel less pain as they don't have a nervous system. Also, invertebrates have a larger surface area to volume ratio, so the caffeine can diffuse through their system very quickly by diffusion alone and doesn't require a circulatory system. So that's something else we need to consider. And that's about it for this test, guys. Don't forget, sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks. If you want me to do more Edexcel AS Biology videos, spread the word, share the walkthrough, and I will bust out as many as I can, but only if they're getting watched. Take care.
You're still here? It's over.